This is the recording for September 12th, 2025, Biology 105. All right, thank y'all for making it to a Friday lecture. It's always a struggle. I know, believe you me. Here we are in week 3, okay? You had your first lab with an actual, with more of a lab activity, right? You had to upload some data that you generated in the lab. Don't forget to watch my dang tutorial video about how to do the analysis, about how to generate the standard curve and use the standard curve equation with Excel. It's a 19-minute video. If you watch it, you'll be done with the assignment by the time the video's over. All right? Cool. We'll have more opportunities to do standard curves, use Excel, all these things. Believe it or not, standard curves are going to be hugely important throughout your work as a biologist. All right? Yeah. And then maybe whatever field you decide to go to, if you don't stick with biology, you'll realize, oh, this is just a standard curve, but they're not calling it a standard curve. Trust me, it'll happen someday. You'll be like, in Excel, I'm making a standard curve, but we're just not calling it that anyway. We've got a week to talk about cells next week, and then we got an exam on Monday of the following week. Okay? So I want that to be on everybody's radar. The study guide is posted, all right, in the week 5 module on Canvas. It's comprehensive. Everything you need to know for the exam is on the study guide. Any questions about that? It's on Mondays when I have it scheduled for. Not next Monday, the Monday after. In week 5, Monday, September 22nd. Cool? Awesome. Any other questions about that? Then let's go ahead and wrap up our discussion. Let's make some serious progress into biological macromolecules so we can talk about the cell. We got so much to talk about. We'll get through all the material by the time it's exam time. Yeah. Okay. So I've been talking to you about basic chemistry, and we're finally getting into biological macromolecules. All right, we're going to take all those rules that we learned about bonds and covalent bonds and polar molecules and charges and all that stuff, and we're going to use it to discuss the major organic molecules that make up a living thing. Those 4 major classes of biological macromolecules are the carbohydrates, the proteins, the lipids, and the nucleic acids. Don't worry, I've got a whole, I've got a whole PowerPoint presentation for each one. Now, there's a standard pattern you're going to see here, right? With these biological macromolecules. It's going to be, there are all of these small monomer subunits, right, that get joined in a chain to make a big polymer. So when you build a protein, you take a bunch of amino acids and you link them together, okay? So you're building a polymer of amino acids to make the protein. The monomer is the amino acid. When building these polymers, there's a standard reaction called dehydration synthesis that occurs, wherein a hydroxyl group from one monomer is removed along with a hydrogen group from another monomer or from the growing polymer chain. That generates a water molecule and forms a new covalent bond. All right, taking off that hydroxyl group right here, taking off that hydrogen means that there's a bond, a covalent electron free here and here, where a new covalent bond can form. Solid? Question, and then I'll get you next year.
So it's not, the bonds aren't formed by water, are they? Or
no.
In there and it gets broken apart when you break from longer to shorter.
When you break from longer to shorter, you consume a water molecule to break them up. But when you go from shorter to longer, a hydrogen and a hydroxide ion are removed, or a hydroxyl group. Yeah. And then they are reacted together. So there's a special enzyme that catalyzes this reaction. Yeah.
Thank you.
No problem at all. Yes, please.
I was about to say, is this the main reason why pretty much all life needs to drink water?
No. No, it isn't, but it makes sense that if you're already an aqueous organism, that you would use water for these things, right? Water's universality in all these different reactions is probably more likely just a natural outcome of the fact that we are water-based life forms because we evolved in the water.
So this is just one. It's not. It may not be the main reason, but this is one of the reasons why we need to drink water, because we need those hydrogen hydroxide molecules to join the monomers to polymers, or monomers to other monomers to make polymers.
It's more that the hydroxide and the hydroxyl and the hydrogen ions or atoms are naturally a part of these molecules, and this reaction happens to produce water, and the opposite reaction happens to consume water. It's not really a determining factor in how and why we need water. It's a tricky hair to split, but yeah. Awesome, good question. Anybody else got any questions about this one? Okay, so we have dehydration synthesis, where water is lost. Hydrolysis, where water is consumed. And you break these bonds. Okay? So how many water molecules would it take to hydrolyze a polymer that is 10 monomers long? Check this out. This polymer is 1, 2, 3, 4 monomers long. If I wanted to break it up, completely hydrolyze it into 4 separate monomers, I would need to break this bond here, consuming 1 water. I would need to break this bond here to consume a total of 2 water molecules, and this bond here to consume a total of 3 water molecules. So if the polymer is 4 monomers long, it takes 3 to break it. If the polymer is 10 monomers long, how many would it take to break it? 9. Exactly right. All right? You don't need to break any, you don't need to do anything on the ends. Cool? Yeah, they're covalent bonds. Good deal. I just want to get into the biological macromolecules categories, honestly. And I couldn't get a damn lick of sleep last night. So I'm, all this stuff that I normally love talking about just feels so uninteresting in this moment. But I'm going to adjust that attitude. Let's go. Here we go. Here we go. We're ramping up. We're feeling the power. Do you feel it? All right. I'm drawing it out of all of you, and then I'm going to return it back to you tenfold. Thank you. Good. Okay. Carbohydrates. We know them. We love them. I wish I could eat only bread. That's all I want, is to eat big loaves of bread and just shove them into my mouth all day. We love carbohydrates, right? They're great. Carbohydrates, what are they used for? What is their structure? Primarily used, when we think about carbohydrates, we're thinking sugars, okay? When we think sugars, we think energy. If you light sugar on fire, it will burn. It contains so much energy, all right? So, carbohydrates, energy storage, makes sense, intuitive. Some carbohydrates are used for structure. The most important one is going to be cellulose, which is the structural carbohydrate used by plants to make their cell walls, all right? What are they made of? Carbon, hydrogen, and oxygen atoms. We're going to see their general formula right here, which is C sub N, parentheses H2O sub N. What does that mean? Here's what it means. NH2ON. So if you're looking at a 6-carbon sugar, then N equals 6. All right? So the equation for a 6-carbon sugar, or carbohydrate, would be C6H12O6. You see, because the 6 goes here, and then is applied to both things in the parenthetical. Cool? All right. So like a 7-carbon sugar would have C7H14O7. That's generally the formula there. It doesn't always hold, though. It's just a general rule. This is like probably the only structural formula, molecular formula you'll have to ever memorize, at least in this class. It's nice that it's no glucose. That's glucose. Here we go. There are hydroxyl groups. There's a carbonyl group in these things. Okay, 2 types of functional groups that you can find in these. Let's take a look at what these carbon structures, these carbohydrate structures look like. Keep the energy building, constantly be building the energy. Carbohydrate nomenclature. OSE, O-S-E. OSE as a suffix indicates that you are looking at a carbohydrate. Back when I was a child, when I was in like 1st grade, Nickelodeon used to do these things where they would do like Health Minute, and they'd tell you a health fact, and they also would stop presenting any content for 2 hours in the middle of the day and say, now go outside and play. There's not going to be anything on TV on Nickelodeon for 2 hours. And that was a thing that they did for us. And that's why I'm so well adjusted, you see. Here's the thing I learned from Nickelodeon. Ose rhymes with gross. And you shouldn't eat too many molecules that end in ose, because those are sugars. That is kind of nice because that ose suffix indicates a carbohydrate. Fructose, glucose, sucrose, lactose, all of these are carbohydrates. They're all sugars, okay? So a triose is a general term for a 3-carbon sugar. Look, it's got a carbonyl group on it. It's got hydroxyl groups on it. C should be C3H, 1, 2, 3, 4, 5, 6, O, 1, 2, 3. It followed the rules and everything. We love it. All right? Trioses. Pentoses are 5-carbon sugars. They follow the formulas. Hexoses are 6-carbon sugars, like glucose. All right, they all end in ose. Cool? All right. If the carbohydrate is big enough, then it can be a linear form and then snap into a ring form. All right? And it'll exist in some sort of equilibrium between the linear and the ring forms. We really are not going to talk about this very much, but for biochemistry or for a better understanding of like metabolisms, it is important. But all I'm going to show you is this and tell you, yeah, sometimes carbohydrates are in a more linear form, and sometimes they fold it on themselves and make a ring form. It affects things like the molecule stability and how they polymerize. It's all very fascinating for a different class. Yes? Hydroxyl and carbonyl. So here's the hydroxyl and here's the carbonyl. OH, yeah. Yep. And yeah, the carbonyl is just an oxygen double bound. Yeah, awesome. Okay, here we go. So when we're talking about, this is more carbohydrate nomenclature stuff, okay? I've already established that the sugars end in OSE, right? Now, we're gonna go from monomers to polymers, and the names that are given for these groups, right? The names that are given are monosaccharides, disaccharides, and polysaccharides. So you've probably heard of a polysaccharide before, and a polysaccharide is just a polymer of monosaccharides, which are sugars. Why are they called saccharides? All right, because of, if I'm not mistaken, it comes from the same root of this glycosidic linkage here. A lot of these polymers get their names from the linkage type. You don't really need to worry too much about that. I'm not going to ask that you memorize the different linkages between the different monomers in the different polymers. I don't think it's an especially useful use of your time, okay? But they have this special type of bond between them. Look at this. It forms via dehydration synthesis. Okay, you take glucose, dehydration synthesis occurs, boom, you get maltose, monosaccharide, disaccharide on the right. Cool? The bond between the monosaccharides is called the glycosidic linkage. I believe I saw your hand first, and I'll get right to you. Yeah.
And disaccharides are only 2 monomers?
Yep. And then, you might say, oh, they're not a trisaccharide. But the convention is once you get up to 3 and above, you just go polysaccharide. But a trisaccharide, you would know what that meant too. It would be 3. Yeah, solid. Question? Oh, look at that. Perfect. So these are the disaccharides. Yeah, they got 2. Here's maltose. In this example here, we've got different monomers being joined. So the monomers can be the same or they can be different, all right? And the polymers get big new emergent properties based on which monomers are joined and in what order. That's how a protein gets all of its important characteristics, okay? Because of which amino acids are joined together and in what order. So here we're looking at a new disaccharide. In this case, glucose and fructose are joined together via dehydration synthesis. All right, establishing a new glycosidic linkage between the 2 monomers, producing a disaccharide called sucrose. There it is. Cool? Disaccharides. Which functional groups have we seen? I put this slide back up because I wanted to just give you a refresher. Like, look, the carbonyl group and the hydroxyl group, we saw them both there, right? Cool? Wait, why is this here? Why did I put this here? I made this big change. I must have thought this was really important. But why would I have put it in the functional group in the middle of... Why would I put it in the middle of the dang carbohydrate section? Great. Well, you know what I've made is a mistake. And you know what that means? That means that I am bad, I am irredeemable, and, class is over forever. What we're going to do is just make a little adjustment on the fly. And I'm just going to try to make it flow. Let's see how well I can do this. Here we go. So we've been talking about the carbohydrates, right? The carbohydrates are a great example of molecules that just have a couple of functional groups. Now, I've only really pointed out to you a single or 2 functional groups present inside of the carbohydrates, all right? Functional groups can give molecules specific characteristics, okay? Maybe you've heard of a sugar alcohol as an example. That should make you think, oh wait, OH groups have alcohols. Interesting. What does this mean? All right, I know there's hydroxyl groups in sugars, But also, if we look at one of these, great, cool. But also, there are many other functional groups that you're going to see in different molecules. Let's talk about them briefly. That was a lame transition, but let's just look at them again, okay? Methyl groups we talked about. I've got these different functions. This isn't good. I don't like this. They're nonpolar. We're going to talk about all this stuff later. Yeah, I hate this. I'm just moving past it. All right, get out of here. Okay, cool. That should have been in the previous PowerPoint, if anything. My most humble apologies. Here we are. We're back discussing carbohydrates. How nice is that? All right, glucose. What's the structural formula for it? There are 6 carbons. There are 12 hydrogens. There are 6 oxygens, C6H12O6. Okay? If you joined 2 molecules of glucose via dehydration synthesis to make maltose, what would the formula be for maltose? This is kind of a little bit of a trick question, right? Because what we've got is C6H12O6 plus C6H12O6 equals what? All right? So how do we do it? What do we need to do, folks? Who wants to put some stuff out there? You got one there? I'll get you next, yeah.
Well, since it's water is being removed, we'd have to assume that H2O is not the carbon.
Yep, so we can go minus H2O. And that is exactly the trick right there. Is it's dehydration synthesis, so 1 water molecule is going to leave, meaning that the formula is going to be C12H22O11. Make sense? Awesome. Great. Was that your point two there?
Me?
Was that, yeah, was that your,
Oh yeah.
nice, awesome. Very, very good stuff. See how that works? So, if you were like, hey, I'm going to join 3 glucose molecules together, what would the chemical formula be? You could do the math in the same way. You just have to know how many water molecules are going to be released by joining 3 monomers in a chain. It gets a little bit tricky. Yeah, please.
One question: is this maltose we're talking about? Is this the same malt that's in like milkshakes and other products?
Yeah. So kind of. Yeah. So when you're malting something, what the heck?
Ground grain.
Yeah. You take grain and you get it malted, right? You're doing that to get it to start to release sugars. And by releasing sugars, that's going to allow it to be like fermented more easily and all that. So this maltose, that's the sugar response that's present there. And then it's like you drink you eat a malted milk ball. And at some point in the long, long past, I'm sure there was malt in the recipe somewhere. But now it's probably a malt flavoring that they make in some terrible lab where they test it by spraying it in like rabbits' eyes and stuff. Because everything's horrible, You can't just have some candy anymore. All right, polysaccharides. So polysaccharides are polymers of monosaccharides. They are joined by glycosidic linkages. What's a glycosidic linkage? It's a bond between 2 monosaccharides. All right, love naming things. Polysaccharides are energy storage and structure, okay? As far as energy storage, the starches are polysaccharides. So plants use starches, like in a potato, okay, to store a bunch of sugar for a long period of time. All right, it's a great way to store energy. You make these special energy storage molecules that are just long polymers of sugars, okay, of carbohydrates. In animals, we also store carbohydrates. We store glucose, okay, in this long polymer called glycogen. It generates glucose, glycogen. It's primarily found in your liver, and this is what insulin regulates, all right, is when your blood sugar gets low, you release insulin, that triggers glycogen breakdown in your liver, and then that raises your blood sugar because it releases glucose into your blood. All right? So starch in plants, glycogen in the liver, serve the same purpose. Energy storage, right? Make sense? Cool, I love it, please.
So are the glycosidic linkages do all? Sorry.
No worries. It gives me a chance to get back to it. What's that?
When polymers are linked together, does every polymer use the glycosidic linkages?
Great question. The glycosidic linkage is specific to the polysaccharide. So you're going to see that every polymer that we talk about has its own specific type of linkage.
How do they differ? Because they all use the dehydration.
They do all use dehydration synthesis. Some of them have an oxygen in between them. Some of them don't, right? But they're named because, they give them a name because that's the bond in that type of polymer, right? So it's a very cyclical definition of like a glycosidic linkage is the linkage that occurs between monosaccharides to form polysaccharides. Yeah. And it will look like this for what that's worth. Yeah. And then, of course, different enzymes can break down different types of linkages, and but they're all covalent bonds that form due to dehydration synthesis. Okay. Also, other polysaccharides are structural. They're involved in providing structure. So we've got cellulose. Cellulose is very important. It's around plant cells, okay? And it gives them structure. There are all these plants, right, that don't have woody tissue, like herbaceous plants, okay? And their leaves can stay strong against gravity, all right? How do they do this? By developing a bunch of internal turgor pressure and then pressing against these cell walls that constrain each cell. So cellulose is a sugar, it's a polysaccharide that's very resistant to degradation that surrounds plant cells. And it's still contains a lot of energy. It just can't be easily accessed because it's not designed for energy storage, right? Now, some organisms can produce enzymes to break down cellulose, but we're going to talk about that in just a little bit. Another structural polysaccharide present in the animals is chitin. Chitin, which forms the exoskeletons of arthropods, right? Fair enough? Good. Starch. Here's some starch granules inside of a plant cell. Check it out, right? It's a big polymer. It's a big old polymer of different sugars, okay? So you break it down, you get the glucose out of it, boom, amylose, amylopectin. There's two types. Pectin has all these branches. It helps to keep the amylose together. Pectin's a general term for things that stick things together. You might have used pectin to make jelly or something. It's like a plant-based gelatin sort of substitute. All right, you don't have to boil hooves anymore, which is pretty cool. You don't need to know a lot about this, but it will become eventually important if you ever go into like biochemistry, the amylose versus the amylopectin. Here's the key thing I want you to know about the starches. The starches are a polysaccharide of glucose, okay? And they can be broken down for energy when it's needed. They're designed for energy storage, okay? So plants produce them when they need to store energy over the winter. And if we munch on a potato, okay, we can break down the starch with enzymes that we have. Fair enough? The structural carbohydrates, oh yeah, I want to talk to you about that. The structural carbohydrates are not designed for energy. They're not designed to be broken down for energy. Check this out. Enzymes are going to be a huge important topic in this course. The amylose, okay, is the name for the big sugar that makes up the starch, the big polymer. Question, please.
So if you're eating like sunflower seeds, are those technically a starch then?
No. The energy store. So when you're eating a sunflower seed, what you are eating are big swollen leaves.
Oh.
All right, those are cotyledons, energy storage cotyledons. Now, there may be a type, as far as the state of the energy storage molecules in the cotyledons of a seed like that, I can't remember 100%. There was a time when I would have had a solid answer for you. But it's not really like a starch. It's not a starchy food. You know what I mean? Yeah.
Would couscous be a good example of a starchy seed?
I'm not sure about that either. Yeah. I just don't know. Yeah. So usually, right, if you're talking about seeds and their energy storage, they use something besides starch. They're using endosperm or long chains of glucose, right? But how much of that is in the form of starch? It's just a question that I don't remember, I don't recall. The starchy things that we usually think about are like potatoes. Yeah. Cool. So check this out, right? Amylose is the sugar, it's got the ose suffix, OSE. Amylase is the enzyme, it's got the ase suffix, ASE. That's going to be a really standard pattern that you see as you move forward in the course. And it's very important, okay? Every time you're looking at a carbohydrate, well, not every time. When you're looking at every time you're looking at a word that ends in OSE, you're probably looking at a carbohydrate, okay? And every time you're looking at a word that ends in ASE, you're looking at an enzyme, okay? We're going to talk about enzymes, but this sort of rule, getting used to these sorts of patterns in science, it makes your life way easier because you can read a word that you've never seen before, all right? And if you know anything about the scientific roots, the word tells you what it is, all right? Like, if you saw something that said, oh, this is a cellulase. Oh, a cellulase. Okay, I haven't seen that specific word before, but I know that it's an enzyme because it ends in ASE. And cellulose is a sugar. So this enzyme cellulase must break down cellulose. What do you think the enzyme responsible for breaking down lactose is called? Lactase, exactly. Patterns like this, it's great. It's one of the good things about science, scientific language, is that, the definition's jammed into the word. Okay, cool. Glycogen, animal storage. Look at this. It's one type of molecule. It's all branched and all this stuff. Okay. Yeah. Stored mainly in your liver. It sort of clumps up and makes little granules too. It can get released. It can be broken down to release glucose into your bloodstream. All right. Cool. Structural carbohydrates. Main structural carbohydrate is cellulose, the main one that we're going to talk about. It's incredibly important. Look at this. You get all these long chains of glucose. All right. So amylose, right, in starch is also long chains of glucose, but they are different forms of glucose. They are different isomers of glucose. And because they're different isomers joined in a different orientation, their stability is drastically affected. We're not going to go into the details of that, okay? But it's important to be aware of that they're both made of glucose, but because they're different isomers joined in a slightly different way, these serve as structural molecules, whereas the other ones serve as energy storage molecules. So here's a plant cell. It's got a cell wall. Animal cells, no cell wall. Bacterial cells, cell wall. Fungi, cell wall. Different cell walls than these cell walls. Cell wall talk done. All right? I'll talk to you more about plant cell walls when we talk about turgor pressure, when you talk about osmosis. But I have a bad habit of getting too into this topic, and I'm going to try to not do that this semester. Unless somebody has a question about it. So check it out. You get these long chains. They're all sort of like wrapped into these cables, and they get laid down on top of each other. It's like a basket weaving situation, and they make a big shell. It's like a leathery shell. Yeah. What about them? They're chitin, right? Anybody remember off the top of their heads? I believe they're based on chitin, yeah? So it comes from, they're more closely related to us anyways. So it's more like the exoskeleton of an arthropod than it is like the cell wall of the plant. Pretty cool. Yeah, fun question. I liked it. Yeah.
So glycogen, it's a sugar.
Yes, it's a polysaccharide, yeah.
And with OSE.
Yes.
So do not all sugars end with OSE. How do you recognize the sugar from the beginning, prefix?
You can't always.
Okay.
You cannot. But the suffix OSE is meant to indicate a sugar. Yep.
So it can be a sugar without OSE, but if it has OSE, it's a sugar.
Exactly.
Okay.
Yeah. So it's nice. Like it's not like every word won't do it for you, but if you see that when you're like glyco, like glucose, like glycogen, it's like that might get you there. I don't know. A lot of times you can guess what a word that you've never seen before is in an article, and you can be right. It's kind of fun. Yeah. Oh, of course. Okay. Cool. So cellulose, it goes around the cell walls. It's super important. Okay. It gives plants their structure. Plant don't have a skeleton. We got skeletons. That gives us our structure. That's an oversimplification, but just bear with me. And have I ever mentioned about my whole no sleeping thing? It's not like a fun thing. It's no good. Here's starch versus cellulose. All right? They have slightly different orientations of these glucose molecules, and these are different isomers of glucose. Cool? So there are ways to think about this. Like, why is cellulose more stable than starch? Well, if you look at the orientation of the hydroxyl groups and these longer chains, then this group and this group won't interfere with each other, and this group won't interfere with each other. But if you look up here, these long chains are all stuck on one side, and that can put an actual physical strain on the molecule, making the glycosidic linkage less stable and more susceptible to breakdown. That is, once again, an oversimplification, but I wanted to show you the actual difference in their structures a little bit. Please. No, the isomer, what I mean is that it's a different it's a different form of glucose here than here. They have different spatial arrangements, right? They're isomers. Yeah. So yeah, this is the beta glucose isomer, and this is the alpha glucose isomer, and they differ in the orientation of these hydroxyl groups. Yeah, awesome. Cool. Breaking carbohydrates down. It's a great way to get energy. We do it all the time, all right? So you can break down the enzymes present in starches, but you can't break down the linkages in cellulose. Very good. Insoluble fiber is cellulose, right? Most of it. If any of you have ever done a low-carb diet before, you know that you'll be reading like the nutrition facts sheet of a food product, and they tell you, oh, subtract fiber from the carbohydrates, right? To get the actual like effective carbohydrates. Well, why? Why would you have to do that? Because cellulose is technically a carbohydrate. So that fiber is technically counted as a carbohydrate. But if it's insoluble fiber, your body's incapable of breaking it down. So you never get at the energy in those bonds, and it passes through your digestive tract. So it doesn't make sense if you're nutritionally trying to keep track of your carbohydrate intake to keep track of all the stuff that you're never going to break down. It's moderately interesting, right? Okay, cool. So cellulose. Some enzymes can digest cellulose. All right? There are no animal enzymes that digest cellulose. Okay? But there are bacterial enzymes that digest cellulose. So some animals survive off of plants where their main energy source is cellulose by having a symbiotic relationship with microbes.